haven't drug-resistant bacteria taken over the world? I'm going to approach that in general, but especially from the perspective of the organism that we study in our wet lab and do a lot of quantitative work on as well, which is Streptococcus pneumoniae, or pneumococcus. So this is work that's funded by a variety of NIH institutes. And it's really, what I'm going to talk about is the result of a kind of slow burn set of projects that have occupied the, kept a number of us awake at night, scratching our heads, trying to understand what's going on for a while, about a decade or so. And these are some of the collaborators on the work that I'm going to describe. So I think it's fairly clear to everyone who works in the field that there is a clear relationship between the level of antibiotic use in a particular geographic region and the amount of drug resistance in that region, where amount is usually measured as a proportion of all cases of a particular infection or infectious agent, or proportion of isolates of that infectious agent that are resistant to particular drugs. And this is perhaps the most classic illustration of that relationship that's shown in sort of every talk in our field, which shows on the x-axis the amount of use, outpatient use of penicillins in various countries in Europe for all causes, for any reason. And on the y-axis, the proportion non-susceptible among strep pneumoniae, the bug that I'm going to spend a lot of time talking about. And several things are really striking about this. One is the variation on the x-axis, which is from the Netherlands to neighboring France. We have about a fourfold difference in the amount of penicillin used per capita per year. And I don't think the French are 4 times sicker, but they, from at least personal experience and reputation, they like to prescribe antibiotics, whereas the Dutch are much more conservative about prescribing. And also on the y-axis, the striking thing is that essentially no resistance at this time. This is now relatively old data. I think that it's from the year 2000. Yeah, 2000. Up to about almost half the population resistant or non-susceptible. So that's a pretty striking pattern, and it's reproduced in other organisms and for other drugs. But this is probably the cleanest relationship that's ever been published, and I must say that if you do any other year in the 2000s, you get a positive relationship, but it's much noisier. So there was some, perhaps, choice of the particular year that was most striking. But there is a relationship, and it's pretty consistent for bugs and drugs. And when you first think about it, you say, okay, that kind of makes sense, that more use is a selective agent for more antibiotic resistance. But if you think about it a little bit longer, it becomes a little bit of a puzzle. And that's because we think of antibiotic use as a selective force, which is something that increases the frequency of a particular phenotype, genetically determined phenotype, in this case resistance, in the population. And there's a counteracting force, which is a fitness cost of resistance, something people have studied in many labs, including ours, whereby resistant strains are often, but not always, slower growing or less competitive in the absence of drug. So in the presence of drug, they're selected for, but in the absence of drug, they're selected against. And that balance creates a net selective pressure for antibiotic, that's the difference between antibiotic use and the fitness cost. But from a basic evolutionary theory, we understand that selection is the rate of change in gene frequency. It's not a steady state. If you select for taller people, it's not that everyone becomes a certain height, it's that the mean height in the population goes up over time, and the same for crop yields or whatever. So selection is kind of the rate of change of the phenotype, not the steady state. And so this leads to what we've started calling the puzzle of coexistence, which is why doesn't drug resistance fix in the population or sensitivity if in fact this net selection is negative? And when my colleague Christoph Fraser, with whom I've done much of this work, said this to a colleague who's a clinician, an expert in antibiotic resistance, he said, and he said we were working on it together, he said, well, that's a very Harvard question. And at Harvard, we're pompous enough to think that that's always a compliment, but I don't think it was actually meant that way. I think it was meant as a kind of ivory tower, who cares, well, let's get on with it kind of question. But I would argue that it's a very practical question. And the reason why is that if you look at the projections on which the current revival of interest in antimicrobial resistance is largely based, there's been there have been little ripples every time a new drug resistance problem arises, but those ripples have sort of died out. And it seems like in the last 3 or 4 years, there's been a sustained amount of attention from world leaders and scientists and funding agencies towards antimicrobial resistance. And this report from Jim O'Neill, funded by the Wellcome Trust and the UK government, is largely, I would say, responsible for that. And the big numbers in that report are very round. They're 10 million excess deaths from antimicrobial resistance by 2050, and a cumulative cost of exactly $100 trillion over the next 35 years. And the scenarios on which that is based are a complete loss of efficacy of essentially all drugs for all bacterial infections. So the idea is that by that time, and there are different scenarios considered, but at least by that time, we will be at a point where no currently available antibiotic is effective against any bacterium. So is that what we're, and these are a little bit more detailed. So this is, there are two different assumptions. One is 100% resistance to all drugs and bugs in 15 years, and another scenario that they consider, and it's not exactly clear which one is producing these, but is a 40% increase in resistance plus a doubling of incidence in all bacteria drug-bug combinations. So either way, it's saying that we are not at a steady state, but we're on the upward slope of a bad trend. So I would argue that that's just not a good description of what seems to be happening in most bacteria drug combinations. These are data from the UK, from another group, not my data, but just illustrating that resistance to penicillins and fluoroquinolones, two of the major groups of antibacterial agents, has been steady in the whole population shown in blue and in various clones over about a decade or a little bit more. Similarly, in fluoroquinolones, there was a little rise, and then the UK, this may be attributable to declines in fluoroquinolone use in the UK, but there's no way that you would extrapolate that for another 15 years and say we're going to be at 100%. That just is not a sensible extrapolation of these trends. If you look at Streptococcus pneumoniae, again, not our data, these are data from Spain, and broken down by serotype, which is a sort of important strain marker that I'll talk about later. Similarly, the prevalence of resistance in different serotypes is shown to two different drugs, is shown in these lines. Ignore the bars. They're shown on these lines, and you see that in some serotypes, there's nearly 100% resistance. In many serotypes, there's 0% resistance, and in some serotypes, there's intermediate levels. And Spain is another of the very high use, at least by European standards, countries. So as a species, there is no way in which you would say this is 100% resistant or going towards that. Recent data from, that I was indirectly involved with from Boston show that in Staphylococcus aureus, another of the important pathogens, first of all, the overall incidence in this Boston hospital is going down, but more strikingly, or equally strikingly, is that most of that decline is from the methicillin-resistant phenotype, the most concerning of the resistant strains, or sets of strains, whereas penicillin susceptible strains are slightly creeping up and are increasing as a proportion. So this is a positive, beneficial trend, not a worrying trend, which is not to say there are no problems. There are lots of problems of antimicrobial resistance, but the scale is one that we need to understand better, and the trend, I think, is, if anything, relatively flat. And that is the puzzle of coexistence. So one possibility for why that is, why we haven't reached 100% resistance in virtually any bug-drug combination, is that we're just going there but slowly, that those are increasing and that they're just doing it very slowly. But an argument against that hypothesis is this analysis of data from Israel. This is antibiotic use of different classes of antibiotics with seasonal variation over several years. And you can see this is a log scale, and so the total use increases in the winter by about a factor of 3, really dramatically increased over the trough in the summer. And that's repeated every year. If you sum over all the years and look at the prevalence of resistance, you can see that there's a repeated annual cycle, that's the average of which is shown here, where resistance peaks when there's a lot of antibiotic use and troughs in the at the end of the summer when there has been little antibiotic use for several months. So it seems that the explanation that it's just very, very slow adaptation is implausible in the sense that the bacterial population can clearly adapt very rapidly to changing amounts of use. So I think that's not a good explanation. Another class of explanations comes out of mathematical models. These are what we call SIR models, although here susceptible individuals are called infected with nothing, or infected with one strain or another, or infected with both. And a number of us had been working with these kinds of transmission models, similar to the ones that Jeff Shaman showed yesterday, but involving both sensitive and resistant strains, for a number of years. And those models can actually reproduce the phenomenon of coexistence of drug-susceptible and resistant strains pretty well, but they do so by sneaking in assumptions that are not biologically justified. And this paper a few years ago laid out what those assumptions are. I'm not going to go into the technical details because it's not the main topic here, but essentially, when you construct one of these models, it's very easy to sneak in by mistake the assumption that sensitive strains compete mainly with themselves and resistant strains compete mainly with themselves, but there's less competition between. And if you understand any basic ecological theory, the idea that within group competition is stronger than between group competition is a recipe for coexistence. But we have no basis biologically to think that's true in the drug-sensitive, drug-resistant world. So if you expunge that false or unjustified assumption from models, you get a more classic ecological outcome, which is that you get competitive exclusion. If treatment is stronger than the fitness cost of resistance, you get the resistant strain reaching fixation, and if the treatment is outweighed by the fitness cost of resistance, the susceptible strain reaches fixation in these kinds of models. And we spent a lot of time trying to make variants of those models with more and more Baroque assumptions. This is one example that we tried where individuals could have double colonization with either strain or double colonization with both strains and various transitions back and forth. And what we were trying to do was find models in which the generic outcome was coexistence. And we did that through parameter sweeps, through various possible values of parameters describing the transmission. And what we found was that there was a region of coexistence here shown in green, where the parameters were kind of tightly balanced with one another. And you could get coexistence, but much more often you got either the resistant strain or the sensitive strain fixing. And so that didn't solve the problem. So in the last few years, couple of years, I think we've begun to understand the solution to this problem, at least in the pneumococcus. And the next little while, I'm going to try to describe what we think the multiple ingredients of that are. And it wasn't one thing. It turns out to be quite a number of things. So one basic premise is that that antibiotic use, and therefore the selective pressure for resistance, is heterogeneous across many dimensions of the population. One is age, in the sense that younger people have more antibiotic use. They carry strep pneumoniae and probably some other pathogens for longer, creating a larger benefit for resistance, and I'll talk about that more in a minute. And also they transmit assortatively, meaning they mix with each other, the young mix with each other and the old mix with each other, and so that amplifies the notion of compartmentalizing selection for resistance or selection for sensitivity. There's also geographic heterogeneity. As we saw, even in Europe, there's fourfold variation in the amount of antibiotic use. And there's also heterogeneity by serotype. I showed you those different serotype-specific resistance patterns and showed you that some were highly resistant and some were not very resistant or had no resistance at all in Spain, and that's repeated across the world, as we'll see. And so this form of heterogeneity also leads to coexistence of resistant and sensitive strains, because long duration serotypes that are typically carried for months get more benefit from being drug resistant. So this is the output of a example model that we published last year, in which we varied along the x-axis are the different serotypes, along the y-axis is the prevalence of resistance, and the duration of the serotypes gets shorter as you go to the right, the duration of carriage. The different colors in the different graphs are different levels of fitness cost of resistance. And what we find is that when we incorporate multiple forms of heterogeneity, including the ones that I described on the previous slide, we do get the phenomenon by which there are some serotypes that are highly resistant, some serotypes that are completely susceptible, and a few in the middle. And which ones those are varies with the amount of selective pressure. But we get kind of the roughly correct pattern, but still a paucity of serotypes for which this kind of within serotype coexistence occurs. But it's on the right track. So I mentioned duration of serotypes being an important cause of this coexistence, and I want to go back to that and talk about it in a little more detail. So these are data from a study in Kenya, an unbelievably detailed study that Osman Abdullahi and Anthony Scott performed, in which they did very, very frequent follow-up of children who were colonized with different strep pneumoniae in their nose, and found that the duration of carriage of this organism ranged from something like over 100 days on average to about 30 days on average. And this is an organism where transmission occurs from people who are asymptomatically colonized. It's part of the microbiome, the nasal respiratory microbiome. It does make a lot of people sick, but a very small proportion of the total number who are colonized. So you can think of it as like an E. coli type thing that's transmitted between people who are not sick from it and occasionally does make them sick. When you compare the serotype, the typical duration of carriage in a serotype with the prevalence of resistance to various antibiotic classes, you find consistent, almost completely consistent relationship between those two in a positive sense. So these are the correlation coefficients of the different relationships of particular drugs with duration of serotype carriage measured in different populations. And we think we understand the reason for that, which is that when you consider antibiotic consumption in a simple model that doesn't permit coexistence, but just of a single strain that's resistant or sensitive, There's a threshold of antibiotic consumption, above which resistance wins and below which susceptibility wins. But the threshold moves depending on the mean duration of carriage. And this is the expression for it, where the clearance rate, which is the opposite of the duration of carriage, has to be balanced by the amount of antibiotic treatment. The reason for that is that if in intuitive terms, is that if a strain is going to be there for 100 days, but every 30 days it gets treated with antibiotics, then resistance provides it, on average, about a 70-day benefit of longer duration of carriage. But if its mean duration was going to be 20 days, and it gets every month exposed to antibiotics, that's a much less beneficial thing because it was going to be cleared anyway. And that comes out of the equations, and this is how it does so. So the idea is that strains with long carriage duration will favor resistance, and strains with short will favor susceptibility. And you can work that out in a model with different costs of resistance balanced by different amounts of antibiotic consumption, and you get this intermediate area where there's a strain that's long duration and is fixed for resistance, and short duration and fixed for susceptibility. So the general idea, which was pointed out by Mike Wade a long time ago now in this article, is that if you have balancing selection on one locus, in this case serotype, which epistatically, that is, modifies the fitness effect of a second locus, the resistance locus, here by modulating duration, that can lead to stable coexistence of alleles at the second locus. So that's the general population genetic point. So we have seen serotype as one of the factors leading to heterogeneity and duration. And previous work that I'm not describing today shows why there's balancing selection on serotype. It's immunologically diverse. It's the major target of immunity, and it's under balancing selection by our immune system. And that's earlier work. But it turns out, based on more recent work, that balancing selection in general, meaning selective pressure that maintains intermediate frequencies of many, many alleles in the population, is really pervasive in this organism. So the graph shown here is plotting the frequency of a particular a COG. So this is a gene in the accessory genome of strep pneumoniae in Massachusetts, in a collection that we analyzed, against the frequency of that same COG or gene, presence absence, in a refugee camp on the Thai-Burmese border called Mela. And what you see is a fairly strong positive correlation, meaning that in these completely unrelated populations, if there's a gene that's at 10% in Massachusetts, it's probably close to 10% in Mela as well. And you could say, well, maybe that's just because it's the same strains that have gone both places, but it turns out that's not true. So if you take a phylogenetic tree and essentially snip off the long limbs of that tree and call each long limb a sequence cluster, so essentially a clade, then the clade frequencies are uncorrelated. So they're common ones in Massachusetts that are completely absent in Mela, and vice versa. So it's not the same strains, but somehow this population assembles itself to collect the same genes at roughly equal frequencies. That's halfway across the world. What if we look at it over time? This is recent work looking at the same thing, but now prior to introduction of a vaccine, and then as the vaccine was introduced, here it perturbed these frequencies because it selectively inhibited certain strains and not others. That the frequencies of the genes were perturbed, and what was striking is that they came back the best predictor of the gene frequencies in the Navajo White Mountain Apache population that we were studying here, of pneumococci that we were studying here, the best predictor of their frequency in 2012 and 2013 was their frequency in 1998, not the intermediates. So it's like the opposite of what you would expect from a random walk. The recent past is not the predictor, it's something in the distant past when the population had equilibrated. So our interpretation is that there is frequency-dependent selection on at least some of these genes, by which if they leave their predetermined frequency, they eventually snap back to that frequency, as shown by the response to this vaccine perturbation over some years. So what we need to maintain this process is both frequency dependence or balancing selection on some locus, and also some genetic determinants of carriage duration. This is a beautiful tree, again, not my work in this case, but John Lee's and colleagues at the Sanger, showing the heritability of carriage duration. So this is how long did the kid in Mela carry the pneumococcus before it was cleared as a function of its genetics. And you can see just from the color coding that there's a lot of genetic determination of duration of carriage, and the heritability, meaning one individual to another who acquires it from them, the heritability of carriage duration is about 0.6. And they find that not only serotype predicts carriage duration as we would have predicted, but also quite a number of other polymorphic genes, some of which are understood and most of which are not, which are determinants, at least by GWAS, by bacterial GWAS, are determinants of carriage duration. So putting all those pieces together, what we think is that the coexistence of resistant and susceptible strains is a fact for large numbers of bug-drug combinations. And that it's a puzzle because the existence of fitness costs doesn't explain it. It should just be going one direction or the other, and it should be doing it quickly based on the results from Israel. Understanding this, we think, matters scientifically and for better decision-making and allocations and policies, and Gila Kaplan's talk yesterday really beautifully illustrated that understanding whether drug resistance is growing, shrinking, or doing both at the same time determines how much you prioritize the dealing with the drug-resistant strains as opposed to all strains. We think that epistasis between resistance determinants and serotype explains some coexistence, but maybe not all of what's observed. We think that another piece of the explanation is heterogeneity of transmission and treatment within and between populations, and that the existence of other loci that affect carriage duration and are polymorphic, otherwise you wouldn't see their effects because it was a GWAS, so they must be polymorphic, and they also affect carriage duration, maybe therefore in epistatic interrelationships with resistance determinants, and that may explain what's left of the puzzle. So I said we were focusing on this bug because it's the one that we've studied, and I think it's the one that's been most studied for this general question. But I think the explanation is always probably going to be multifactorial. So in this case, beware of Occam's razor, you can cut yourself. I think in this case we really have lots of different factors that are contributing to the coexistence of resistant and susceptible strains. How will this play out in other bugs? Coexistence as an observation seems to be the rule, as I suggested in the earlier slides. I think other bugs, we have some ideas about other factors that may be important. For those organisms that receive most of their transmission and a lot of their antibiotic exposure in the hospital, earlier theoretical work by us and others suggested that that the heterogeneity between the hospital and the community may maintain coexistence for a while. I think there are some examples, for example, fluoroquinolone resistance in the agent of gonococcus in Asia has nearly fixed or fixed in some places, so coexistence is not true everywhere and for every bug. And another explanation may be feedback. If doctors are observing the levels of resistance and reducing the use of drugs to which an organism is resistant, that could, in principle, lead to a steady state. The evidence that those changes happen fast enough is limited, and many factors promote multi-drug resistance, whereby changing drugs doesn't necessarily reduce selection for the old resistance, it just selects for a strain that's resistant to everything. And recent work by Sonia Leighton that builds on the work that I just described suggests that that will be a generic feature because of this feature of selective benefit being tied to duration of carriage. And there may be others. So I'll stop there and again thank this large group of collaborators and take your questions.
Questions?
Yeah, so I was wondering whether there are other factors that one could consider that could also explain this passing observation, and that we see, for example, in the resistance in cancer cells. It's a totally different setting, but at least two factors were novel to us, and that's non-genetic persistence and persisters, and the other is community effects of populations. So can you comment on that, whether that also contributes to this counterintuitive finding?
Sorry, what do you mean by community effects?
Interactions between individual cells, bugs, that support each other.
I see. So in terms of non-genetic resistance and persisters, I think that's clearly a real phenomenon, and that the coexistence of persisters with the sensitive cells, I think, is reasonably well understood. I can't think of a way in which this phenomenon is that, because it's clearly genetic resistance. We know the genetic determinants. But it would be interesting to think about whether there are ways in which persisters could somehow buffer the sensitive strain against clearance, and so that would be a good idea. In terms of community effects, there have been suggestions that, for example, certain bacteria secrete beta-lactamases that they produce and thereby protect their neighbors. I think that's not a general explanation, in part because it doesn't explain target change or other mechanisms by which there's not an obvious interaction with the neighbors, but it could be a particular piece of it.
Thanks. So oftentimes when you see coexistence in nature, you have competing interactions, meaning that you have, for example, one part of it's more stable, one part of it has higher proliferation or what have you. But you can see these kind of things all through nature in terms of, I don't know, length scales that emerge, coexistence of different populations, what have you. It goes to your notion that there's not an Occam's razor way to think about this, because there's multiple explanations, but they're in balance. So I'm just a physicist, and so I think in terms of competing interactions, in terms of generating populations, such as what you're talking about. But I don't know if that's relevant here or not.
Yeah, I mean, I think that's kind of where we started and then got stuck and decided that wasn't a good enough explanation for this. I agree that, I mean, the paradox of the plankton is a real thing. It's been remarked on for decades in biology, and coexistence of lots of things is the rule. And in some sense, we think of this as an interesting model system to study it. The reason why just sort of saying, well, it's what happens is kind of unsatisfying and positing competing interactions is that resistant and susceptible bugs, co-circulate in the same community. The same hosts have them, often in sequence or sometimes together. It's just not, there's no niche separation that we can see except for the fact that antibiotic treatment selects for the resistant strains and fitness cost selects for the sensitive strains. So it's
Well, I'm not urging that you, okay, so for example, in tumors, one can have a very highly heterogeneous cell mixture, some of which are really proliferative, some of which are drug resistant. And initially one might think, well, why don't the drug resistant ones win out? Or why don't the more proliferative ones? And they don't, and it's because there's a balance. There's simply a balance in those two. And if you quantitate that balance, you recapitulate the population that you observe. Does that make sense?
So it makes sense. I think spatially, a tumor that's got spatial structure and limits on proliferation and all those things, all of that can oppose the survival of the fittest, to put it in crude terms. But when you have transmission across different individuals,
No, that's true. That's true.
it's just
Yeah.
the models say you get competitive exclusion, and unless we put in every one of these bells and whistles, generically we get competitive exclusion. There is a region of parameter space, but it can't possibly include both the Netherlands and France.
What about diffusion? So I'm thinking in terms of how you deliver a drug, right? So like in the case of pneumococcus, or you're going to be, your drug is going to be delivered to your bloodstream, and then whether or not the gradient for drug delivery to different parts of your lung is based on where the capillaries are, et cetera. So like the idea that some sort of like environmental niche in various parts of the lung could definitely be playing a role here.
Yeah, I think within an individual that makes sense, although what's weird about the pneumococcus is that as best we understand, the population that transmits, and that's therefore of evolutionary consequence, is all in the nasopharynx. The lungs are, which is, it's still some heterogeneity,
Yeah.
and maybe just distance from the mucosa or whatever. So I think within an individual you could imagine that, but the evidence is pretty limited still, but probably the transmission inoculum is a few bacteria, not a whole population. But it does help. It helps a little bit in these models if you allow co-colonization with sensitive and resistant strains that are both transmitted simultaneously. That does help.